morning, class. So, in today's botany lecture, we are going to see a new plant that is Marsilea, and we are going to see at what places Marcelias are present, basically their habitat, their morphological structure means their outer structure, and as well as their anatomical portion means we are talking about their internal structure. So, all these three major issues we are going to see about our plant Marsilia. Marsilia also known as a water fern because most of the pteridophytic plants, as the Marsilia is a pteridophyte, and most of the pteridophytes they live on the terrestrial plant. They lived as a terrestrial plant, or you can say they lived on the land. But some of the plant, like every division has some exceptions, so the are terrified, and they are having the exception in the name of having the Marsilia. So Marsilia is a bottom fern, and now we are going to see the normal features about the Marsilia. So if we say that the commonly it is known as a paper bot, or you can say the paper fern, because pteridophyte also known as a fern, as because of their ferny structures, or you can say the fern look appearance. We can call them the fern, but it is not a normal kind of fern. You can say because the normal kind of fern they are present in the terrestrial or the land plant, but it is the one which is present inside the water. So that's why we can say that they can also as known as a water fern. Although it is fun, so it is represented by about like 53 species. So as Marsilia is a genus, and we studied plant taxonomy, and we know that genus contains several kind of species in them. So here, this genus Marsilia, they are representing like 53 species worldwide. Means in all over the world, only 53 species of this genus are present, and they are also in cosmopolitan. Means they can be present everywhere, but the most the place where they abundantly found and mostly found that are the subtropical zone or the countries like Africa or Australia. So in Africa and Australia, mostly all the species, all the almost all the species of Marsilia has been found, and even in India, we have the nine species reported by the genus Marsilea. So, if we see their systematic classification here, so we will be starting from their kingdom. Kingdom is Plantae, clade, Trachyophytes or Pteridophyte division, Polypodiophyta, class Polypodiopsida, and order Salvinates, family Marsillaceae, and genus Marsilia. So this is the genus we are studying. Actually, when we study some kind of plant, mostly we study them to their till their genus level, because genus is a point where most of the species which come under that genus have almost similar character, only having some differences or variations. Otherwise, all the features are mostly similar. So here also in the genus of Marsilea, most of the species which come under the Marsilea have the common kind of characteristics. You can see how beautiful this plant look, and they are the mostly you didn't notice, but you have seen this plant in the ponds or the lake, around the lake. Mostly they are present in our India also. In even UP, Bihar, we got these plants. Either the species are hydrophytic or amphibious. So there are two conditions when we are talking about the Marsilea habitat. Means hydrophytic, they can live inside the water. And second condition we are talking about here, that is amphibious. Means they can be possessing two kind of lifestyle. They either be inside the plant, or sorry, either be inside the water, or they can or be present on the land. When a species or when a genus possessing two kind of lifestyle on a land, on a water, we can call them the amphibious. So they grow rooted in mud or marshes, so they can be rooted, means their root can be present inside the muds and marshy area, like some swollen pool or something which is watery but not exactly like a water body, but yes, they have more composition of water in their mixture. So marshes, shallow pools, or completely submerged or partially or entirely out of the water in wet bodies. So these are the conditions in which our Marsilea plant can be present. Like we are talking about here, completely submerged means completely under the water, surface of the water. Partially means half of their body will be above the water surface and half body will be down to the surface. Entirely out of the water habitat means total body of the mycelia will be on above the surface of water. Marsilea hirsuta is Australian xerophytic species. So, as we know that the Marsilea is a kind of a species, water loving, but still everything have exception like we talk about. So, these Australian xerophyte species, Marsilea hirsuta, that is a special kind of exception because Marsilea do not face water deficit condition. And when we are talking about the xerophyte species, they are the kind of species which has to face the water deficit condition and low amount of water for their survival. So this kind of species is unique. Marsilea hirusta and Marcella quadrifolia are two most common Indian species. So if we are talking about what are the common kind of Marsilea species, quadrifolia and hirusta, and this is also the one which is a xerophyte. Next is here, invasive species usually found growing in marshy places means all other species, if we don't consider exception Hirusta, all other are placed inside the marshy places, wet soils, near muddy margins of pond and are commonly found in UP, Punjab, Bihar and Delhi. So this area is preferable, or you can say they are having the favorable kind of conditions to grow Marsilea there. Now here, this is the structure of Marsilea. You can see these are the leaves. Okay, these are not flower. Okay, they look like very beautiful flower, but they are not flower. They are the leaves here, and flower cannot be present because it is a pteridophyte, and flower is a characteristic feature of the angiosperms. And as they are pteridophyte, they do not possess any kind of flower arrangement. So these are the things which you see, they are the leaves. These you can say they have very long petiole. This the one which is attaching them. They are very long petiole and rhizome-like structure. Rhizome inside mostly when we are talking about the aquatic plant, root and shoot. They have little combined structure. They are not very clearly differentiated between the stem and root. So that's why this rhizome there is like the complete structure having the shoot plus root, and then we are having this root. Root are a separate, but the rhizome is considering as the shoot plus root condition. Also, they are having this sporocarp condition is here. The sporocarp, you can see that they are very small, tiny organisms which are going to have the spores production inside the mycelia. So they are responsible for having spores inside them, and they are responsible for the reproduction of mycelia. Now we are going to see some external features, the things which you are seeing in this diagram. Now we are going to see them one by one. External features of Marsilea. So the mature sporophyte is a herbaceous plant, and its underground rhizome is spread in a diameter of 25 meter or more. So you can see how much diameter they can possess by their rhizome. The plant body is distinctly differentiated to rhizome, leaf, and sorry, rhizome, leaves, and roots. So these are the three structures which can be present inside the mycelium. Wait. So this first we are going to see in this whole pattern or whole structure. We are going to firstly see the rhizome. Rhizome is like a shoot kind of a structure here. So all the species possess a rhizome which creeps on just behind the soil surface. So they just present or creeps just beneath the soil surface here. It is cylinder, dichotomously branched with distinct nodes and internodes. So if we see their portion, that how the rhizome is look like, so they are the majorly the cylinder portion dichotomously branched and with distinct nodes and internodes. And it is capable of indefinite growth in all direction, as a result of which is occupy an area of 25 meter or more in diameter. So it is cylinder dichotomously branched. So it is very it is very easy to be for their transporting system. Transportation is can be very easy by having this kind of conditions in. So this cylinder and dichotomous branch means they can have these two branch pattern inside them, or their nodes and internode are capable for having the indefinite growth. So indefinite growth means they are not something which has to be very limited growth pattern or something up to a limited growth speed. They can be like in very broad category like 25 meter or in more. In aquatic species, the internodes are long, while in subterrestrial species they are short. So in aquatic species, the internodes they will be very long as comparison to the subterrestrial species. So what is a subterrestrial species? Means the thing which are present on the land, but they are having some partial water condition also. So usually from the upper sided nodes, the leaves are given out, while the lower side the roots. So on the leaves, or you can say the upper side of the nodes, when they are having this nodal portion, so on the above side of the node we are having the leaves here, but at the downside of the node we are going to have roots. So now we will going to see the leaves here. So I think the rhizome structure is clear. They have indefinite root, they have nodes, they have internode, and in subtropical subterrestrial they are having the short internode. In the aquatic one they are having the long internode. Leaves, they are born alternatively on upper side of rhizomat node. So we just take it out from the rhizome that leaves had to be present on the above or the nodes. Young leaves show succinate venation like ferns. Succinate venation means they having some fold situation. When leaves can be fold just to increase, I think I have a diagram here. Here we go. So the leaves, when they are having this, a quite fold in them, so they can be called as a succinate venation, and this is a characteristic feature of the fern. In some species, young leaves are covered with multicellular hair. Some of the species of the Marsilea, they also have these multicellular hairs in them. The leaves are compound with basal petiole and terminal lamina. So these are not simple; they are the compound kind of leaf. They are having this basal petiole, very long petiole, and terminal lamina also. Terminal lamina means the surface area like this. This is called the lamina, and these are the petiole. In submerged plant, the petiole is very long. So as we just seen in the diagram that the plant having a very long petiole and very flexible. So they can be very flexible. So this flexibility and longness, they help plant to float over the surface, because as much as water they can be dispersed or displaced, the more they can swim on that. So that's why this flexible and petiolic structure is very long, and that is a cause that they got a buoyancy over it. Lamina floats over the surface of water, but in muddy or marshy plants, the petiole of the leaf is short and rigid, with short lamina spreading in the air. So if we see the condition on the terrestrial plant, so terrestrial plant they do not have any kind of situation for swimming of these lamina. In that case, petiole will be short and they will be rigid. They are not like very flexible and very soft structure. They has to be rigid because they needed to make the plant to look straight. So that's why they had it to be rigid. Short lamina spreading in the air. Short lamina has to be spread in the air. The lamina consists of four leaflet. As you've seen, there is a four leaflet present on a particular lamina. This is called pinnae. Four leaflet situation. They are known as pinnae, which are present at the apex of the petiole. So we've seen that they are present at the last portion of the petiole. The four leaflet arise as a result of three dichotomies of the lamina in close succession to each other. So the four leaflet they arise as a result of three dichotomies of the laminas and close succession to each other. So they will be like presenting very close to each other in the four number three or very closely each other. Two leaflet arise slightly higher than other two. So in whole dichotomous arrangement in this, when we are talking about a one pinnae, so in one pinnae, two leaflet arise a little higher to the in comparison to the two other ones, because this highment can keep them into the balancing inside the water also or in the land situation also. Now, some scientists which work on the leaf pattern here, Puri and Gurk, suggested that the leaf consists of single pinna consisting four pinnules. Okay, the leaflet which we are talking about, the Puri and Gurk, they tell them or they give them a new name, that is the four pinnules. So pinna have got a dichotomously branched vein system with cross connection. The pinna which we have here, they have dichotomous branch vein system and in the cross section. So cross section and dichotomous section, why it is obtained here? It is obtained here to maintain their curriculum. So maintaining their curriculum, transporting the things which are required, because leaves are the one who is going to do the photosynthesis and they are going to make amount of the food material. So that has to be secreted throughout the plant, and for that purpose they have this vein system cross sectionally connected. So the vanillets at the margin are connected with loops, thus forming a reticulum, and the shape of the pinna varies from ovate to obensate, and margin also varies from entire to crenate or crenate to lobed. So crenate and lobed kind, this is the structure that a pinnule is going to have here. They can be little ovulate or ovate in situation. Margins also can be like sometimes they can be very crenate and sometimes they can be lobed. So this can also vary from species to species. Sometimes the pinna are once and twice deeply dichotomous lobed. So sometimes it can be twice deeply dichotomously lobed. A two-toed. Two-toed means one on the overlapping situation with two. In minuta, Marsilea minuta, this kind of situation has been seen. At night, the pinna are folded upwardly. This is known as sleeping movement of pinna. So, in the night portion, what they can do, the pinna folded upwardly in upward condition, not downward condition. It is upward condition. So, this is known as sleeping movement of pinna. Basically, this thing happens for some excess amount of water if in aquatic thing. So in aquatic field, maybe leaves is stomatas can absorb more water, so that's why they just fold it out. Near the base of petiole, the stalk bean shaped sporocarp are born. So near the petiole stalk, so where the petiole is attached to the rhizome, at their stalk portion, bean shaped sporocarps are born. So the reproductive units they will be going to place at the bottom of petiole and the origin of the petiole from the rhizome. Now the third structure which we are going to see here, that are the roots. So roots here, they are the adventitious arising from the upside of the node of rhizome and either singly or in group. So their root can be, single in structure, group in a structure, and sometimes they can be given out from the internodes also. So that depend like aquatic plant. In the most of the aquatic plants, root are not as much as they has to be developed because root. What is the main function of root? Root is present to absorb as much as water they can do, but plant is already present inside the aquatic nature and they do not need that kind of water absorption because they are already present in water. What are their major concern to be there? The major concern was there for obtaining the oxygen for their survival. So that's why the root do not has to be very developed here, and because they already present inside the water. Now, this was the whole structure of the outer structure. Now, we are going to talk about internal structure of Marsilea. So, whenever we are going to, study about the internal structure, there will be the aperture we are going to take for studying the internal structure. That has to be a TS or a transverse section. So what we are using here, the TS of first, that is rhizome, or you can say the stem of the marsupial. So TS of the young rhizome here, what we used, that young rhizome, they show the protostelic structure. Protostelic means their steel should be protostelic in nature. Pith is absent here. These are the common features we will going to see one by one in outline diagram. Pith is absent. Okay. Xylem is completely surrounded by phloem, but in old stem, pith is developed in the center, and stele is amphipholic hypnostele type. Means in the young phase, their pith is absent. The stele is also protostelic, but as the plant got through their maturity or the plant just evolving, they will be having the pith, and also they are changing their stelic portion from primitive protostelic to the developed amphipholic sepnostelic type. So this kind of changes that can be seen inside the Marsilea from their young stage to their developed stage. So here the tears of fold stem. This is somewhat circular in outlining and show the following features. So when we see the tears of old stem here, young stem features I told you it will be not there, and steel was protostelic. Otherwise, all the things are the same as we were going to see in the old stem. Epidermis, the first thing which we always study in the anatomical portion because they are the one which is the outermost layer and the covering layer. So epidermis is outermost limiting layer and single cell thick parenchymatous cells, and the stomatas are absent. So stomatas, they do not need to be present here because transpiration rate will be very slow, or you can say if they are present on the surface, the transpiration rate will be very high. But in the rhizome, there will be no stomata present. Stomata will be present on the leaf things. The epidermis, that is outermost limiting layer of single cell and thick in parenchymatous. So parenchymatous layer is in epidermis. Cortex differentiated into the three regions. So you can say they have a like massive kind of cortex which has to be distributed into the three zones. So three zones which we are talking about here: the outer cortex, middle cortex, and the inner cortex. So we will going to see one by one how the outer, middle, and inner cortex look like. Outer cortex is present just below the epidermis, and sometimes they can also be called as a hypodermis. So it is parenchymatous. Okay, when we see their feature also, so they are exactly same like the epidermis. They are also the parenchymatous, and maybe one or several cell thick. So they can be one cell thick or they can be several cell thick structure. Some of the cells contain tannin, so some of the cell from these cortex, what they contain tannin inside them. And tannin, you can say there as a storage material. Middle cortex, it is called parenchyma. Now, this is an important feature that has to be seen under any aquatic plant. Presence of the parenchymas. Parenchymas are the air chambers because, like, if the plant is present on a land, the major concern of these kind of plants is to get the water. But if the plant and you are if inside the water body, what will be your major concern? Your major concern will be getting air. So the same thing happens to these plants also. They also need air for their respiration, their photosynthesis. They needed air. So for getting air, or you can say they got air, but they also store air for their, like, some situations, for unfavorable situation. As a normal plant store water, they store air. So this so large air specks or chambers, they are cell-thick parenchymal septa. In the xerophytic species like the Anupriya, the air chamber are obliterate. So basically, if we are talking about the xerophyte species, means which live in a water deficit condition, they do not possess air chamber because they do not need it, because they do not live inside the water. But if the plant is you can say hydrophytic or water-loving plant, they will need the air chambers inside them. Inner cortex, it is solid tissue of several cell thick. Outer layers of the thick wall is sclerenchymatous, and while the inner layer of the cell thin wall, that is parenchymatous, are compactly arranged. So their chamber, they have these two layers. Okay, the outer layer is sclerenchymatous and the inner layer of having the parenchymatous cells. And compactly arranged, some of these cells are filled with starch or tannin also. So why this sclerenchymatous zone are present here? What is the need of having this sclerenchymatous zone here? So basically, the working of sclerenchyma is to give the mechanical support, and that is this sclerenchyma tissue. They are having very thick wall, and because of having thick wall, they are almost dead cell. Almost mean they are dead cells, and because of these dead tissues, what dead tissue always do? They give the mechanical support. So that's why they are present here to giving the mechanical support to the plant. Now the steel. Steel is amphiopholic steel in the mature plant. In the center, and there is a pith which may be either parenchymatous in aquatic species or sclerenchymatous in the terrestrial muddy species. So that pith, they can also vary on the basis of aquatic and terrestrial species. Like if we are talking about aquatic, there will be a parenchymatous, but if we are talking about a xerophytic or water deficit or terrestrial, they will be having the sclerenchymatous pith. Xylem is present in the form of complete ring, which is surrounded by both served by a complete ring of inner and outer flooring. So you can say the bicollateral kind of xylem phloem present here or vascular bundle here, in which xylem is present in a center, and from both the side, from the outer side and from the inner side, they are having the phloem to them. You can see the was the radius of the petiole, I think. Yeah, just don't have the diagram of this diagram either. All other we have. So basically, in this condition, that is, the xylem will be present in center. Both of their side will be present of the phloem tissue. Pericycle will be there, and endodermis also present. I think you know the endodermis. This is the last layer of the. This is the last layer present of the cortex. In this way, the continuation of the different tissue in the form of complete ring and a stele as follows. The first thing, outer endodermis, when the stellic region start. The very first thing, outer endodermis, outer pericycle, outer phloem, then xylem, then inner phloem, then inner pericycle, then inner endodermis, and this in this way the whole stele has been designed. All the things are in a duplex form here except the xylem. you has to be considerate because everything in a second form, but not the xylem. Why? Because xylem is a water conducting tissue. As the plant is already present inside the water, they do not need xylem for transporting water, because in every place of the water, in every place of the plant, water is already present in an abundant situation. They do not need it in a very higher amounts. So that's why this xylem, they do not need to be present everywhere. So that's why they are just present in a single form here. Now the second thing in an anatomical portion, we are going to see the TS of petiole. So petiole, they are somewhat circular in outline and differentiated into epidermis, cortex, and stele. So these three kind of composition can be seen inside the petiole. So this is a diagram. You can see the outer layer of the petiole. They are consisting the epidermis cells. Epidermis, you can see the compactly present parenchymatous cells. They are present. Just after that, we are having this outer cortex or hypodermis. The same structure what we see in the rhizome. They're not much different. Then you can see these large air chambers. Middle cortex we talk about now, that we are having this large air chambers for storing the air, because this is the major concern of hydro plants, that air should be present in the inside. So that's why this air chambers are present here for storing the air. After that, what we are having here, we are having the pericycle and inner cortex just after the middle cortex, and when the inner cortex will be ending, we are having this endodermis. After endodermis, we are having the pericycle, we are having the metaxylem, protoxylem, and phloem here. Phloem, metaxylem outer side, protoxylem here, and this is our phloem. So these are the situations, or these are the anatomical portion that are present inside the TS of petiole. So the epidermis, outermost layer of single cell thickness, and the cell are parenchymatous and slightly elongated. That what just we seen in the diagram. Cortex, same as was in rhizosperm. They are also divided into the three regions: outer cortex, middle cortex, and inner cortex. Outer cortex just present below the epidermis, and you can also say they can be called as hypodermis, and that will make up of the parenchymatous. Middle cortex, we talked about that they are present for having the air and chyma inside them, or having the large air chambers. So these air chambers are separated by single layered partition of thin walled parenchymatous cells. Inner cortex, this is solid tissue of several cell thickness, and these cells are parenchymatous, contains large and tannin-filled cells. So after the parenchyma chamber, we are having inner cortex, and the cells of inner cortex, they are filled with the starch and tannins, because they are the storing part of plant. Minuta, fused sclerenchymatous layer are also present just inner to the middle cortex. Some plant like Minuta, Marsilea minuta, they have this sclerenchymatous zone there, and I told you what is the function of sclerenchymatous zone to be present to give the mechanical support to the plant. Steel, it is somewhat triangular in the outline and is of protostelic type. Okay, so steel of the petiole, this is not the steel like the rhizosphere. They are having the steel is a very primitive kind, that is protostelic. And pith is absent, pith will be absent. Xylem is V-shaped. The shape of the xylem will be the V shape with two distinct arms. So the two arms will be present. When we see the shape of V, there are like two arms condition. Each arm is provided with metaxylem element in the center, and protoxylem is situated at both the margin. So in this case, you can see protoxylem is exarch in condition and metaxylem always in endarch in condition. So in this way, they can be present. The xylem is surrounded on all sides by phloem. So if we see the xylem condition, they will be surrounded by the phloem. Phloem is externally surrounded by a single layer of parenchymatous pericycle, that is, which is turned is bounded by a single layer of endodermis. So having the centrally placed xylem, after xylem we are having phloem. After phloem, we are having the pericycle, and pericycle will be bounded by the endodermis. So this is the, you know, this is the layer formation that can be seen inside the petiole. Now, what we have to see, we have to see the TS of leaflet. That is the third structure of the plant. So, same, all the key layers will be same like the epidermis, cortex, on inner steel, but their compositional analysis inside the epidermis, cortex, and steel they can be different. So, here the epidermis, it is the outermost surrounding layer and it is only one cell in thickness, but it will be whenever we talk about the epidermis of leaf, there will always two kind of epidermis. One is the upper epidermis, second is the lower epidermis. In floating leaflet, the stomata are present on the upper epidermis, but in case of plant growing in mud, moist cell, so basically if the plant is aquatic, then stomata will be present on the upper surface of the leaf because they are exposed to the sun. And in this way only stomata can do the function for the transpiration. But if the condition is terrestrial or xerophytic, then situation will be opposite and stomata will be present on the lower side because they at that time they cannot expose themselves for the over excited transpiration rate because it will lose immersed amount of water from the plant, and in terrestrial plant we cannot face that kind of situation, but yes, in the aquatic plant we can do as much transpiration as plant can tolerate. The mesophyll, and in now also if we see that inside the leaf there is also a second difference, that in place of cortex we are having the mesophyll tissue. Mesophyll tissue is the combination of two kind of special tissue that is called the palisade and spongy. Always in upper portion, palisade tissue is present. In the lower portion, we will be having the spongy tissue. And palisade and spongy tissue have a special function to play that is called the photosynthesis because they are the unit which are consisting having the chloroplast inside them. And because of having so many chloroplasts inside them, that's why they are going to do the function for the photosynthesis and making food material for the plant. This is a situation or this is an anatomical portion of the leaflet. Like first and second, you can see upper and lower layers epidermis. After the just epidermis, we are starting having this, you know, mesophyll tissue in the form of upper epidermis, just behind the epidermis, the palisade one. And after this, we are having this spongy tissue, and at the lower portion, we are having the parenchyma because this is also important. We needed to store air because photosynthesis needed air. Even in the case of making food for CO2, carbon dioxide, and when they are going to make the oxygen, that is also be stored in these kind of air and chyma. So the both the things which has to be secreted by this bundle, that is a centrally placed one single vascular bundle will be present here, in which phloem is outer and xylem is at the center. So in this way, the internal structure of leaflet has to be look like. Then the upper and lower epidermis, palisade and spongy tissue, and just behind the parenchyma for storing the air. So vascular bundle, in between the mesophyll tissue are present several vascular bundle. Each vascular bundle is concentric and amphifibril type, means made up of a centrally situated xylem surrounded by all side by a phloem. So you can see only center we are having xylem and that will totally covered with the phloem tissue. Xylem, we don't need much amount xylem, that's why it is centrally placed, but we need phloem here because phloem are the transporting agent for the food transportation. That's why we need phloem here, but we do not need xylem in much amount. Next, TS of the root. So TS of root is somewhat circular in outline and can be differentiated into the epidermis or piliferous layer, cortex, and stele. So now we can also see that their outline also differentiated in the epidermis, piliform layer, and cortex and stele. This is somehow diagram of internal structure of root can be seen. You can see the epidermis. Epidermis is a very primitive first layer. After that, we are having outer cortex, and in place of middle cortex, what we are having the same parenchymas. Then we are having very internally placed inner cortex, when this, and after that we are having pericycle, and the last layer of the cortex will be endodermis that is present. After this endodermis, we are having pericycle. After the pericycle, we are having these vascular bundle in our center. So epidermis is simple, outermost parenchymatous single layered covering. Cortex that is differentiated into the two part here. Not sorry, three part. There are just two parts here, outer and inner. Outer cortex is like consisting of whole air chambers here, and basically the inner cortex. What we are talking about, inner cortex that is differentiated into the outer parenchymatous and inner sclerenchymatous zone. Outer cortex is totally, you can say, totally responsible for collecting the air. Inner cortex is dividing in two layers. Inner cortex, first the parenchymatous zone and inner layer. In the inner cortex, in the inner layer will be sclerenchymatous, which is giving the mechanical support. So, in this way, the inner cortex is delimited by a single layer thick endodermis. So, last limitation layer of the cortex will be the endodermis. Steel, if we see the root steel, it is protostelic type, simple, primitive kind of species that the same kind of species has been seen in the leaflet. Occupies the central position in the center. It is devoid of pith, means pith will be not present. Xylem is situated in the center, which is diarch and exarch. Means there will be two xylem bundles that will be present, and there will be exarch in condition, means towards the outer side. It is surrounded by the phloem, and they will be surrounding by the phloem also here. And the phloem is bounded externally by a single layer of pericycle. So in the center we are having the xylem, which is diploxylic, or sorry, not diploxylic, diarch in condition, means two xylem bundle in the region towards the outer side, exact condition. But that whole portion will be surrounded by phloem, and phloem will be surrounded by the pericycle. In this way, the stele has been designed in the root of Marsilea genus. Okay, that's all about the Marsilea morphological structure, their habitat, how they look, and how they internally look. So all the internal aspects, external aspects, their habitat we has been completed. But still, if you guys have any problem, you can go through these references about the Marsilea, and you will going to have more knowledge on different aspects of Marsilea. And still, you have any problem, you can ask. That's all for the today.